Okay, so for all you listeners out there, the ones who really love to get into the nitty-gritty of ideas, well, today we're going deep on a concept that's like the bedrock of biology. You know what I'm talking about, right? The genome.
Yeah.
You hear it everywhere these days, health, evolution, even when people talk about like what makes you you.
Yeah.
But for folks like us who are used to really picking apart these big foundational ideas, the question becomes, what is the genome, really? I mean, seriously.
It can get pretty complicated, that's for sure.
Right. There's just so much info out there now and all these debates within genomics and postgenomics, and it can feel like you're drowning in it. So what we're going to try to do today is cut through all that and give you like a clear path through it all, with a bit of a philosophical lens.
Exactly.
So for this deep dive, we went all in on the genomics and postgenomics entry from the Stanford Encyclopedia of Philosophy.
The beast.
Right. Like seriously in-depth, academic, all that.
Really gets into the weeds on the terminology, the history, and the philosophical implications of all this genetic stuff.
Yeah, which should appeal to our listeners, right?
Yeah.
The ones who like to get philosophical.
Definitely.
Okay, so our mission, if you will, is to try and unpack this whole genome thing. Like, what does it actually mean? How has that meaning changed over time?
Yeah.
And we're going to touch on some of the really interesting philosophical debates it sparks and the goal. Well, we want to give you a nice, concise overview of it all.
Great plan.
So let's get started. I guess the logical place to start is with the gene itself, right?
It makes sense.
Like, where did that idea even come from?
So officially, the term gene was first introduced in 1909 by Wilhelm Johansen.
No hat.
Yeah. He used it to refer to these units that are responsible for passing down traits, like, from parents to offspring, the stuff of inheritance. Then, about a decade later, in 1920, we get the term genome. This one comes from a botanist named Hans Winkler.
Ah, interesting. So a botanist came up with the genome. Okay, what was he thinking?
His original definition is actually pretty interesting. It kind of shows you how things were thought of back then.
Okay.
So he defined the genome as the haploid set of chromosomes.
Haploid, meaning...
So haploid means just one set of chromosomes, like what you get from one parent, not the full paired set you get from both.
Okay, right. I remember that from, like, high school biology.
And he said it was the haploid set of chromosomes in conjunction with the associated protoplasm.
Protoplasm.
Yeah, basically all the stuff inside the cell that surrounds the nucleus. He saw this whole package as the material basis of what he called, it's usually translated as a species or systematic unit. So right from the beginning, the genome was thought of as something material, something physical that was working within a specific context inside a cell.
Huh. So even back then, it wasn't just about the information. It was about the physical stuff, too. Okay, so we've got gene, we've got genome, but the whole field of genomics came much later, right?
Yeah, much later, yeah.
Like, when did that term even pop up?
Genomics didn't really appear until 1986.
Wow, okay.
It actually came up during a meeting where a bunch of scientists were trying to come up with a name for a new journal.
A journal, huh?
Yeah, they wanted to start this journal specifically focused on sequencing genomes, mapping them, comparing them, all that. Thomas Roderick, a guy from Jackson Laboratory, he's the one who suggested genomics. He was basically making an analogy to the field of genetics, which was already established. But the funny thing is, that was actually the first time that omics suffix was used like that. You know how we have proteomics now and metabolomics and all those?
Right. Ocomomics everywhere these days.
It all kind of started with genomics.
Okay, but the weird thing is, nobody really knows where that suffix came from.
It's a bit of a mystery, yeah. Even now, the exact origin and the original meaning of that is unclear.
So we know where the words come from, but like you said earlier, the way we understand genome now, it's not so straightforward.
It's gotten messy.
Yeah, it has. When I was reading some of those definitions from really reputable sources online.
I know, right?
It's like, hold on a second. There's some fuzzy thinking going on here.
It does make you think.
Definitely.
It highlights some of the conceptual problems.
Yeah.
Like, if you look at the definitions from places like genome.gov, the U.S. National Library of Medicine, Nature's Education Portal.
Big names.
Yeah, exactly. You see this pattern where they kind of mix up the information the genome carries with the actual physical stuff it's made of.
I see what you mean.
Like, for example, the National Library of Medicine, they define a genome as an organism's complete set of DNA, including all its genes. And then they say it contains all the information needed to build and maintain that organism.
All the information.
Right, and that's where the philosophical alarm bells start going off.
Because
Yeah.
it's not all the information, is it?
Exactly. It completely ignores things like the environment, the stuff the mother's egg contributes during early development, even the symbiotic relationships that are, like, crucial for so many organisms to survive.
Right, like gut bacteria, that kind of thing.
Exactly. If all the information's in the DNA, does that mean a symbiotic bacterium's DNA becomes part of the host's genome?
Whoa.
It creates these weird boundaries, you know?
Yeah. So it's not as simple as saying it's all in the DNA.
And then you get genome.gov. They describe the genome as both a set of instructions and the 23 pairs of chromosomes.
Okay, so they're saying it's both the information and the physical thing.
Yeah, like they're the same thing, which is kind of tricky. You're merging this physical object, the chromosomes, with this abstract idea of instructions.
It feels like they're trying to fit two very different things into the same box.
It is. And that, I think, is a key takeaway for us, for anyone trying to think about this stuff. When we try to define something like the genome, something so fundamentally biological, using language that kind of blurs the lines between the physical and the informational,
We run into trouble.
we do.
Yeah.
It highlights this really deep philosophical problem in even grasping what it is we're studying.
Makes sense. And then there's this idea, this really simple idea, that the genome is just all the genes an organism has added together.
Yeah, that one's tempting, but it doesn't hold up.
Right.
Even if we ignore how hard it is to even define a gene, we know that in lots of organisms, humans included, a big chunk of the DNA doesn't code for proteins. It's non-coding.
It's not making genes.
Right. And if the genome was just the genes, then whole genome sequencing wouldn't make much sense, would it?
Because you're sequencing the whole thing.
Yeah, all the DNA in the chromosomes, not just the bits that are genes.
Okay, so the simple definitions aren't working.
Yeah.
So where do we go from there? Like, how do we define this thing? I know the Stanford entry suggests 2 starting points.
It does. So the first one is to say the genome is the sequence of all the nucleotides.
Nucleotides, that's like the A, C, T, Gs, right?
Exactly. Those are the building blocks of DNA. So the genome is just the order of all those letters. This definition might include DNA that's outside the nucleus, like in the mitochondria.
Right, mitochondrial DNA.
Yeah. And the main reason for this definition is that it focuses on the information, like the sequence is the code, it tells the cell what to do. The second approach is to say, no, the genome is just a physical object. It's the chromosomes themselves, and that's it.
Okay, 2 very different ways of looking at it. So the first one, the genome as sequence, it kind of makes sense at first, but then you've got this whole field of epigenetics, right?
Yeah, and epigenetics throws a wrench in the works.
It does. Like, it says the sequence isn't the whole story.
Exactly. Epigenetics looks at how gene expression can change, even if the DNA sequence itself doesn't change. And what they found is that there's all this stuff happening on top of the DNA sequence that affects which genes are actually turned on or off.
Okay, so it's like there's a control panel that tells the genes what to do.
Kind of, yeah. You've got modifications like DNA methylation, where these little chemical groups attach to the DNA.
Okay.
And you have histone modifications, which are changes to these proteins that DNA wraps around. All of this stuff plays a huge role in which parts of the genome are actually used.
So the sequence is like the blueprint, but epigenetics is like the contractor deciding which parts of the blueprint to actually build.
Yeah, that's a good way to think about it. So the sequence represents the potential, but the epigenetics determines what's actually happening. The nucleotide sequence, even though it seems stable, it doesn't give you the whole picture of how the genome actually behaves in a cell.
Fascinating. So that's why the article leans toward the second definition, right? The one that says the genome is the chromosomes themselves, the physical stuff.
Right, because it's not just the DNA molecule itself. It's also all the stuff associated with it, like the histone proteins and their chemical states, all those modifications. That's part of the information too.
It's all packaged together.
Yeah. Like, take methylation, for example. If you see a C in a DNA sequence, it's supposed to represent cytosine, right?
Right.
But how do you show when it's 5-methylcytosine? It's not just a regular C anymore. It acts differently.
It's a modified C.
Exactly.
Yeah.
So if you're just writing out the sequence as a string of letters, you're missing that crucial information.
It's like a hidden layer of code.
It is.
And on top of that, the genome does more than just store information. It has all these other jobs in the cell. It's not just a passive library.
Totally. It's becoming clear that the genome as a physical structure does all these things that go beyond just coding for proteins, like its organization in the nucleus, how it's folded and arranged. That's super important for controlling gene expression. It's also involved in the physical mechanics of the cell, like how it divides and maintains its shape.
Wow, so it's like a scaffold, a framework for the whole cell.
It is. And the chromatin fiber, which is the DNA and histones together, that acts as a scaffold for a bunch of proteins and even internal membranes. So if we just define the genome as the sequence, we need a whole new term for this physical structure and all its non-genetic roles.
Okay, I'm seeing the problem with just the sequence idea, but what about that idea from Dawkins, the whole immortal genes thing?
Yeah.
Doesn't that support the sequence being the most important part? It seems like it's saying the sequence is what endures, it's what gets passed down through generations.
It's a compelling idea, for sure. This notion of genes being these almost eternal things that drive evolution. But even if individual genes were kind of immortal, the genome as a whole, it's constantly changing.
Really?
Yeah, it's dynamic. It interacts with the environment, with the cell, and it's always adapting.
Okay, so it's not just a static blueprint.
Not at all. Even the DNA sequence itself, which seems so stable, it needs constant maintenance. There are all these repair mechanisms that fix damage and keep the sequence intact. It's more accurate to think of the genome as a process, a constant interplay of molecules that keep the organism alive and functioning.
So it's not just this unchanging essence, it's active, it's responsive.
Right. And importantly, it's a two-way street. The organism also works to maintain the genome, keep it stable. They depend on each other.
It's a partnership.
Yeah, exactly.
Okay, so the very definition of the genome is still up for debate, and it's got these huge implications for how we understand biology on a really fundamental level.
Definitely.
All right, let's move on to reading the genome. This whole endeavor started with viruses, right? Like, that's where we first figured out how to sequence a genome.
You're right. The very first genome to be sequenced was a virus, a bacteriophage called Phi-X174.
Phi-X174.
Yeah. It was sequenced by Frederick Sanger back in 1977.
Okay, 1977.
It was a huge deal. It showed that we could actually determine the complete genetic code of an organism. After that, people started sequencing other viruses and then some simpler organisms like E. coli and the nematode worm C. elegans.
But the big one, the one everyone knows about, is the Human Genome Project.
Absolutely. The HGP was a game changer.
Yeah.
Officially, it ran from 1990 to 2003, and it was just massive. Not just because the human genome is so big, it's about 3.2 billion base pairs.
That's a lot of base pairs.
But also because it was this huge international collaboration. They had all these scientific and technological goals.
So it wasn't just about getting the sequence.
Not at all. I mean, getting the sequence was what captured everyone's imagination. But a lot of people would argue that the technology they developed and the other organisms they sequenced were just as important, maybe even more so, in the long run.
Interesting. So what were people saying back then leading up to the HGP? Was everyone on board?
Oh, there were definitely critics. Some scientists were worried about the funding. The HGP was a huge project, big science. And some people argued that it was going to suck up all the money that could have gone to smaller, more focused research projects.
I can see that being a concern.
Yeah. And then there was the whole junk DNA issue. People were saying, why sequence the whole genome when a lot of it seems to be useless? Why not just focus on the genes and the parts that actually do something?
Because that would be cheaper and faster.
Right. But the third criticism, that one was the most interesting, I think, philosophically speaking. It went after this basic assumption of the whole project, that if we knew the entire DNA sequence, then we'd automatically understand how our bodies work, how diseases happen, and we'd be able to cure everything.
So it's like having the sequence doesn't magically solve all the problems.
Exactly. Critics were saying this is just sequence reductionism. You're underestimating how complex biology really is.
Yeah, because there's more to it than just the genes.
Exactly. And that criticism has stuck around, especially when we talk about common diseases. Like even now, we're finding that common genetic variants can't fully explain why people get sick.
It's a web of factors.
It is. And it raises some interesting questions about how science works. Like, should we go for these big, broad projects that try to describe everything? Or should we focus on smaller, more targeted experiments that test specific hypotheses?
Two different approaches.
And then the criticism about sequence reductionism, it gets to the heart of how hard it is to understand complex systems. You can't just look at one layer of information and think you've got it all figured out.
It's about the interactions.
It is.
So looking back, it seems like some of those criticisms are valid. People were definitely oversimplifying things, thinking the sequence was the magic bullet.
Yeah, but the HGP also led to some surprising benefits that nobody really predicted.
Like what?
Well, being able to compare genomes across different species, that's been huge for understanding evolution.
Makes sense, seeing how the genomes have changed over time.
and it's become super important for forensics, using DNA to solve crimes.
Right, right. Okay, so it's had these unexpected impacts. And there's this philosopher of biology, Sarah Richardson. She makes this interesting argument.
Oh, yeah. She says that maybe the real value of genomes isn't that they're like the ultimate cause of everything in biology, but that they give us this incredibly precise way to manipulate organisms and cells.
To intervene. Okay, so the HGP wasn't just about getting the human genome. I mean, there isn't just one, right?
No, there isn't. Every person, except for identical twins, has their own unique genome.
So much for the human genome.
Yeah, the HGP actually produced a kind of composite reference sequence. They used DNA from a bunch of anonymous donors.
A mixed bag.
Right. But besides the reference sequence, there were 3 other really important outcomes. The first was that they deliberately sequenced a whole bunch of model organisms.
Model organisms, meaning?
Like organisms that scientists use a lot in research because they're easy to work with.
Got it.
So they sequenced bacteria, like Haemophilus influenza, yeast, that's Saccharomyces, the Cerevisiae, another bacterium, Escherichia coli, the nematode worm Caenorhabditis elegans, a plant called Arabidopsis thaliana, and the fruit fly, Drosophila melanogaster.
Wow, that's a whole zoo.
It is. And this comparative approach, looking at all these different genomes side by side, it's been incredibly valuable for understanding these basic processes that are shared across all kinds of life.
Right, so if you see the same genes or similar sequences in all these different organisms, it probably means it's doing something really important.
Exactly.
Okay, so comparative genomics. What was the second big impact?
The second one was technology. The HGP needed such advanced tech to do all this sequencing that it really pushed the field forward.
Like they had to invent new stuff.
Yeah, they had to. And this led to the development of what's called next-generation sequencing, or NGS.
NGS.
NGS is way faster and cheaper than the old methods.
That's always good.
It is. And that made it possible to sequence DNA from all kinds of new sources, and it led to entirely new fields of study, like metagenomics, which we'll talk about later.
Okay, NGS is a big deal. And the third big outcome from the HGP?
The third one was that we learned a ton about the structure of the human genome itself. Like we found out that humans have way fewer protein-coding genes than we thought.
Mm-hmm.
It's only around 20,000.
20,000?
Yeah, way less than people were expecting.
Huh.
And then we found out that a huge part of the human genome, about 45%, is made up of these repetitive sequences and transposable elements. Those are also called jumping genes.
Jumping genes. Why are they called that?
They're sequences of DNA that can actually move around to different places in the genome.
Wow. So it's not just this fixed string of letters. There are parts that are like mobile.
Exactly. And this ties back into the whole junk DNA debate and the puzzle of genome size.
Right, because if so much of it is repetitive or these jumping genes, what's it all doing there?
That's the question. So the HGP, it was amazing, but it actually made some of these fundamental mysteries about the genome even deeper. It showed us how much we still didn't know.
So we got the sequence, but we still don't fully understand what it all means.
Exactly. And that led to new approaches, like the HapMap project, which focused on understanding all the variation within human populations, not just one reference genome.
The HapMap project. Okay. And that leads us nicely into the C-value enigma and this whole concept of junk DNA. This area is full of these philosophical head-scratchers, isn't it?
Oh, yeah. The C-value paradox, it's been around since the 1950s, and it basically says that there's no clear connection between how complex an organism is and how big its genome is.
It's counterintuitive. You'd think more complex organisms would need more DNA.
You would, right.
Yeah.
But, for example, some types of onions and amoebas, they have genomes that are way bigger than the human genome. Onions, yeah. And they don't seem more complex than us, at least not in terms of how their bodies are organized.
Right, right.
And even within groups of similar species, like buttercups or some single-celled algae, you can see huge differences in genome size.
Yeah.
Doesn't make sense if you just go by complexity.
So something else is going on.
Yeah, definitely. And this discrepancy is what led to the term junk DNA. It was coined by Susumu Ono back in 1972.
Susumu Ono, okay.
His idea was that the number of genes should basically increase with genome size. Like since mammals have way bigger genomes than bacteria like E. coli, we should have millions of genes, right?
It would make sense, proportionally.
But even early on, people were finding that mammals had way fewer genes than that.
Mm-hmm.
So Ono said, okay, that means most of the DNA in mammals must be non-functional. He called it junk.
It's just taking up space.
Basically. And this mismatch between the number of genes and the complexity of an organism. It's sometimes called the G-value paradox.
The G-value paradox. Okay, so the HGP, with its finding that humans only have about 20,000 genes, that didn't really help with this paradox, did it?
It didn't. It actually made it worse. Some people thought that non-coding DNA would solve the C-value paradox. They said, okay, bigger genomes just have more non-coding DNA, that's all. But a philosopher of biology, T. Ryan Gregory, he argues that it actually creates a C-value enigma.
An enigma, okay.
Yeah, because now we have these new questions, like where did all this extra DNA come from? And what's it doing there if it's not junk? Gregory thinks we need a more flexible approach. He says that different types of non-coding DNA probably have different origins and different roles. Some might be functional, some might actually be junk, but we don't know for sure.
So the HGP didn't solve the mystery, it just made it more interesting.
I think so.
All right, the article then moves on to the post-HGP era and talks about the International HapMap project and metagenomics as two big examples of where genomics went next.
Yeah, those were big.
So let's start with the HapMap project. What was that all about?
So the International HapMap project started in 2002, and it's this huge effort to map out the common patterns of genetic variation in humans.
Variations, meaning?
Meaning differences in the DNA sequence. Like between you and me, there are millions of spots in our genomes where the letters are different.
Millions.
Millions. But they were specifically focused on these things called single nucleotide polymorphisms, or SNPs. SNPs are single-letter changes in the DNA, but they have to occur in more than 1% of the population to be considered a SNP.
Okay, so they're not just random mutations, they're these common variations.
Right. Now, any two unrelated humans, their DNA sequences are about 99.9% identical.
We're all very similar.
We are. But that tiny 0.1% difference, that still translates to millions of SNPs scattered throughout the genome.
So we're mostly the same, but those little differences make a big difference.
They can, yeah.
Okay, and these SNPs, they're not just randomly spread out, are they?
No, they're not. They tend to be inherited together in these blocks.
Blocks, like chunks of DNA.
Yeah, and this is called linkage disequilibrium, or LD for short.
LD, okay.
It basically means that SNPs that are close together on a chromosome, they tend to be inherited as a unit, like a package deal, from parents to kids.
So it's like those genes for red hair and freckles you mentioned earlier.
Exactly.
Yeah.
They might be separate genes, but they're so close together on the chromosome that they usually get passed down together.
I get it.
These linked SNPs, these blocks, they're called haplotypes.
Haplotypes.
So the HapMap project, they were trying to figure out what the common human SNPs were, how often they showed up in different populations around the world, and how they were linked together in these haplotypes.
That's a lot of information.
It is.
So why was it so important to figure out these haplotype structures?
Two big reasons. First, it gave us a more efficient way to study how genetic variation affects human traits, especially diseases.
Like how genes influence whether someone gets sick or not.
Exactly. Because SNPs within a haplotype are usually inherited together, researchers could focus on studying just a few key SNPs called tag SNPs instead of having to look at every single SNP.
Tag SNP, so they're like representatives of the whole haplotype.
That's it. And this made something called genome-wide association studies, or GWs, possible.
GWs.
With GWs, you could scan the genomes of lots of people with and without a particular disease and see which SNPs, or really which haplotypes, were associated with the disease.
A shortcut to finding disease genes.
Exactly. It was a big deal because back then, sequencing a whole genome for every single person in a study was still way too expensive.
Okay, so that's one reason. What was the second big implication of the HapMap project?
The second one was that because haplotypes can be different in different populations.
Right, so a haplotype that's common in one group might be rare in another.
Exactly. So the HapMap project had to get DNA samples from a diverse group of people all over the world.
To get a global picture of human variation.
And that brought up this really tricky issue. Race.
Yeah, that's a minefield.
It is. See, after the HGP, a lot of researchers thought we were going to finally be done with the idea of race as a biological category. Like the HGP showed that humans are so genetically similar, 99.9% the same.
Right, so we're all basically the same. Race is just a social construct.
That was the hope. But then, when they started analyzing genetic variation in projects like HapMap, they found that there were some geographical patterns, like certain allele frequencies were more common in certain parts of the world.
So there were clusters of genetic variation that kind of lined up with continents or regions.
Right. And some people said, hey, this means there is a biological basis for race. These classifications, They're useful because they tell us something about underlying genetic differences.
Okay, so that was the argument for a biological basis of race.
Yeah, they said it could be useful for things like figuring out disease risk or how people will respond to different drugs.
Personalized medicine.
Exactly. But a lot of researchers pushed back against this view. Hard.
Yeah, that was bound to happen.
They said, first of all, these geographical clusters of genetic variation, they don't really match up neatly with the racial categories we use in society.
Right, like those categories are kind of arbitrary anyway.
They are, and they pointed out that human genetic variation is mostly clinal. That means it's like a gradient, you know? There aren't these sharp dividing lines between groups. It's more like a gradual shift in allele frequencies as you move across geographical areas.
It's a spectrum, not separate boxes.
Exactly. And different traits, both genetic and physical, they don't always change together in the same way across these gradients.
So you can't just use one gene or one trait to define a race.
Right. And then there were methodological criticisms. People said the way you collect samples and the statistics you use can influence the results and create artificial distinctions.
So the way you analyze the data can bias the outcome.
Yeah. There's this program called Structure that's often used in these studies, and people criticized it because you have to tell the program beforehand how many genetic clusters you're looking for.
Ah, so you're kind of baking the answer into the analysis.
You are.
So this whole debate about race and genomics, it really shows how hard it is to separate science from our social and cultural ideas.
It does. It shows how deeply intertwined they are.
Okay, let's move on to the second big post-HGP development, metagenomics. Now, this sounds seriously cool.
Metagenomics is pretty amazing.
Tell me about it.
It's also called environmental genomics or community genomics, and it's all about studying the genetic material from a whole community of organisms, not just one species.
A whole community, like in the soil or the ocean.
Yeah, or even the human gut. They take a sample from the environment and analyze all the DNA in it.
All the DNA from all the different microbes living there.
Exactly. And the key thing about metagenomics is that you don't have to culture the microbes in the lab first.
Culture, meaning?
Meaning growing them in a petri dish.
Ah.
But the thing is, most microbes, like 95 to 99%, they can't be cultured in the lab at all.
Really? Wow.
Yeah, it's called the Great Plate Count Anomaly.
The Great Plate Count Anomaly. Okay, so metagenomics lets us look at all these microbes that we couldn't study before.
Exactly. It opens up this whole hidden world of microbial life.
That's incredible. So what about the name? Where does that meta prefix come from?
It actually has a few layers of meaning. First, it means that it goes beyond the limitations of culturing. Like, we don't need to culture the microbes to study their genes.
Okay, so it transcends the old methods.
Second, it means we're looking at the whole community, the aggregate